Alright, so today's video is about polymerase chain reaction, or PCR. We're going to explain what PCR does and get a better understanding of it. So we have a double-stranded piece of DNA here, and we want to make many copies of the part that's in the red box. Now to do that, remembering that double-stranded DNA is part of your chromosomes, and that this sequence is going to be unique. So there'll be one, or in the case of a diploid cell, two copies of this within the cell. To even know whether it exists or not, you're going to have to make multiple copies to amplify the original signal to be able to see whether this piece of DNA that you're looking for actually does exist within the cell. We want to amplify what's in the red square, that piece of DNA. So this is, let's say, 300 base pairs. And we want to make many copies of this double-stranded DNA. Now to do so, to make many copies of this segment, aka to clone it, DNA polymerase is used. Cloning just means that you're making many copies that are identical copies of each other. For DNA polymerase to make a new DNA strand of DNA, it needs a template and a 3' end to add nucleotides to. Each strand of double-stranded DNA above will be a template for DNA polymerase. So we have template 1, which is the top template, 5' to 3', and we have template 2, which is the bottom strand, and that will be 3' to 5', because DNA and double-stranded DNA, they run anti-parallel to each other. But you can tell that these are complementary, right? There's a T, there's an A, there's an A, there's a T, etc. But there is not a free 3' end for DNA polymerase to use to make a new strand. Because what DNA polymerase will do is it will come and look at this and go, okay, there's an A here on my template. Now I'm going to put a T down here. There's a T here. I'm going to put an A. There's a C here. I'm going to put a G, right? But it needs a free 3' end to make a new strand of DNA. Same thing with the other strand. To provide a free 3' end for DNA polymerase, primers are designed and used. So here's again our sequence, and we're going to go ahead and highlight the sequence in green. So this green part is what we want. We don't want the stuff that's in black. If we separate those two strands to get our two templates, so what we can say is that this thing in the red box here is going to be my forward primer, and this thing in the box right here, the red box, is my reverse primer. Now, what we're going to do is for the forward primer, we have here 5', TACG, TAGC, TAAATC, right here, 5' to 3'. And the reverse primer is, because we're going in the other direction, 3' to 5', we have 3' TACG, CATGG, CATT, TA. Okay? Now, a PCR reaction mix will contain the following ingredients. Template DNA in nanogram amounts. You want very little bit of template DNA. Equal amounts of both forward and reverse primers. These are going to be in micromolarity amounts. Deoxytriphosphate nucleotides. These are the monomers DATP, DTTP, DCTP, DGTP in equal amounts. Taq DNA polymerase. Taq DNA polymerase is only active around 72 degrees Celsius. It's a special type of polymerase. And a PCR cycle consists of the following steps. You have a denaturation step, which is at 90 degrees and 95 degrees Celsius. Annealing of primers, which is at 40 degrees to 50 degrees Celsius. An extension by Taq polymerase at 70 to 75 degrees Celsius. And then you go back to the denaturation step. So basically, once you have all the ingredients in your tube, all you do is you're changing the temperature. Everything else will take care of itself. You start at 90 to 95, you go to 40 to 50 degrees, then you go to 70 to 75 degrees for some amount of time for each step. Now, here's again our piece of double-stranded DNA, and the green is what we want to amplify, clone. We do this denaturation step, which is at 90 to 95 degrees. The two strands will come apart because of the amount of heat that's being applied to them. Decreasing the temperature to 40 to 50 degrees will allow the primers to attach. So you can see this is our reverse primer. Remember, the reverse primer was this thing right here, but this thing right here is complementary to this thing right here. And the forward primer sits right on the bottom right here to the bottom strand. you'll increase the temperature to 70 to 75 degrees, and that will allow the Taq polymerase to extend. So you can see that now it has a free 3' end. Let's go back. You can see that there's a free 3' end here. There's a free 3' end here that DNA polymerase can work with. So it goes ahead and extends in this direction for this strand, and it extends in this direction for this strand, for strand 2. And you have two results. These two things are different for each other. 1 and 2 are different from each other. Results from the first cycle are already different than the original template. We will follow template 1 through the second cycle. So we're going to follow number 1 through the second cycle because it's gone through one cycle now. We're going to go through the second cycle. And the results for template 1 will be similar to template 2. They relatively speaking, it'll be similar results in the end, okay? But we're just going to follow template 1 just to show what happens and once you go through cycle, another cycle. Now, why it's different is because you can see that in 1, you don't have that stuff hanging off this end, okay? Right here, with the red on the right-hand side. And of course, with 2, you can see you have nothing hanging off on this side. So you're kind of already cutting off and making your cloning that segment that you want. Now we've taken 1, we go through the denaturation step, 90 to 95 degrees, separate the strands, decrease the temperature to 40 to 50 degrees, allow the primers to attach. You have these free 3' ends again. You increase the temperature to 70 degrees, or 70 to 75 degrees, which is ideal temperature for Taq polymerase, and you get extension on both ends. Again, the results are different than the first cycle and the original template. And now what we're going to do is we're going to follow 1'. Instead of 2', we're going to follow 1'. Okay, so this is the second cycle completed. And 1', we go through the same thing, denaturation. You separate the two strands from each other, decrease the temperature to allow the primers to attach, and then extension again. And you're going to get extension, and you can see here, we've made a blunt end for number 2' right here, right? There's a blunt end already, right? All the extra stuff is gone. Now we're going to follow 2' through to the next cycle. And you can see that, okay, 2' is going to go through separate the two strands from each other. But when you heat it up to 90 degrees to 95, decreasing the temperature to allow the primers to attach, extension. And now what you have is, if you look at 1', you just have that segment. You no longer have any of the extra stuff at the end. Now, for any of the other iterations that we didn't follow, it will all end up in the same place where you have just that area that you wanted to clone and nothing else. All the extra stuff will be gone. So this is 3 random base pairs. And what you can do then is you can run your results after you run your sample and do PCR. You can run the resulting reaction mix on an agarose gel. Now, what does an agarose gel do? An agarose gel is going to separate DNA based on size. Because this is only 300 base pairs, it will run at a certain speed through agarose. Agarose is just a medium, okay? Imagine that if I had a very viscous solution and I put stuff of different sizes on the top of it and let it go through by gravity. Stuff that is bigger will move slower and stuff that is smaller will move faster just because of the amount of friction that is being applied to it. So you can see here that in this picture that I have of a sample agarose gel, that the big pieces of DNA, like 7 kilobases, that's 7,000 bases, is running much slower than something that's 1,000 base pairs. So you run your sample on a gel, and then you can see that, oh, I have this nice tight band showing me that I got the result that I was asking for. I cloned that piece of DNA. And that's the power of that. Now, what can the PCR be used for? To search for the existence of a sequence within the genome, to clone multiple copies of a gene or a segment of a gene to be used for further experiments. Example. So again, cloning. I know this gene exists. I'm going to clone this gene, make so many copies of it, and then I can use it to further manipulate that existing gene by adding sequences to that gene, like mutating that gene. Using it as a tool for genetic engineering, for example, removing the wild type copies being the normal copies, and then adding the gene back in, but under the control of a different promoter, such as an inducible promoter. We'll talk about this a little bit later. PCR in this format is not quantitative. And we'll talk about why it's not quantitative a little bit later because we will talk about quantitative PCR. It simply gives a yes or no answer to whether a sequence exists. It cannot be used to compare the amount of the original sequence in different samples. Okay guys, we'll stop here and continue later. Thank you.